Okay, so again, thank you for coming, because you've been here for 3 weeks now doing quite a tough course and also doing quite tough modules now. So for those of you who are doing the module that I'm teaching on, some of this will be repetition/revision, and also for those of you that are doing the course that Jan teaches on, where he's covered something about instrumental variables, again, it will be some revision of that. When we teach an introductory course on Mendelian randomisation in the unit that I'm from, we do at least 3 days, full days. When we're doing it to a group where it's completely new, then we would do a week. So if you've never done any Mendelian randomisation before, the expectation is not that you'll go away and understand it from this. The hope is that you'll understand something a bit more and be aware of some of the pitfalls. There's lots of things I could have put in this talk, which is not possible to do in an hour, and I'll try to highlight some of those as I go through. The main thrust of the talk is, after I've just gone through what instrumental variables are, and Mendelian randomisation is simply instrumental variable analysis with genetic variance, the main thrust of the talk is about two-sample Mendelian randomisation using summary data. And I'll explain why I think that is important, and it's because I think that that approach is going to become more and more and more common and potentially affect clinical guidelines and public health practice. And as with most methods, it's open to all sorts of misuse/abuse. And even if you think you're never going to use this method at all, it might be prudent for you to understand some of those pitfalls and to be able to read papers and make sense of whether what the people are doing is appropriate or not. And part of the reason I think it's going to become more and more used is you could walk out of this room and do a two-sample Mendelian randomisation with aggregate data in anywhere between 30 minutes and 2 hours. You might not know what you're doing, you might make mistakes, and you may interpret it wrong, but certainly you could do it with the publicly available data. Okay, so just to start, I'm going to start by talking about instrumental variable analysis, and particularly just emphasize that all that Mendelian randomisation is instrumental variable analysis where we use genetic variance. But if we take a step back and think about why we might want to do that, it's a different approach to conventional ways of trying to find out if something is causally related to an outcome than, for example, multivariable regression, where what we try to do in multivariable regression, if we're interested in whether blood pressure causes coronary heart disease, or more specifically interested in quantifying a causal effect, where that could be anything from a minus to 0, null effect, to a positive, what we would do with multivariable regression is try to adjust for as many confounding factors as we can in our data and hope that we're doing a good job there. The reality is that we're likely to have residual confounding, either from unmeasured confounders in our data set, so we can't control for them, or because some of the measures that we are able to control for we don't have measured well. So we might have smoking measured simply as yes, no, ever smoking or not, as opposed to a more refined measure of pack years of smoking over the life course. The other thing that can be problematic is what's often called reverse causation, but is another form of confounding by prevalent disease. So either if people have early stages of disease and it's not known about and not diagnosed, or if you have early stages but you haven't removed those from your analysis or removed the early phases of follow-up, you can have prevalent disease related to your risk factor, and obviously is going to be related to you being identified as having that as an outcome. Again, it's sort of compounded by prevalent disease, or the same as reverse causality. There is another way that we could use to examine this question of what's the causal magnitude of blood pressure on coronary heart disease, and that's using a randomised control trial analysis. And in that approach, what we would do is an instrumental variable analysis in randomised control trials. So rather than being interested in whether antihypertensive drugs are effective in reducing coronary heart disease, what we would be doing here is taking randomization to hypertensive treatment, yes or no, and using that as an instrumental variable to test the effect of blood pressure on coronary heart disease. And this is a sort of classical directed acyclic graph for an instrumental variable analysis. What the graph is showing you is that you don't think it's plausible that there is a link between being randomized to antihypertensives and either measured or unmeasured confounding factors. That you think that randomization to antihypertensive is known to be causally related to blood pressure, or is at least plausibly causally related to blood pressure. And lastly, as there's no arrow going from randomization to hypertensive through to coronary heart disease, you also assume that other than through its relationship to blood pressure, this instrumental variable, this randomization to blood pressure treatment, is not related to coronary heart disease other than through its effect on blood pressure. So those are the three assumptions of this particular instrumental variable analysis. They're also generalizable to all instrumental variable analysis. So for this example, I would argue that I'm confident, let's assume that these are all well-conducted randomized control trials. I would argue that this is going to be true, that blood pressure lowering treatments are going to relate to blood pressure level. That the second assumption, that this randomization to blood pressure treatment is not going to be related to measured or unmeasured confounding of blood pressure with coronary heart disease. Again, I'm convinced that's true if you've done a well-conducted randomized control trial. So we're left with this last assumption, which is randomization to take a hypertensive lowering, a blood pressure lowering drug, yes or no, is only going to be associated with coronary heart disease through its effect on blood pressure. And that's the one that, in this approach, that's the source of bias that we are likely to worry about most. So in the conventional multivariable regression analysis, we're worried about residual confounding. If we're using an instrumental variable analysis in randomized control trials, what we're worried about is this last assumption, that there might be a path that goes from randomization to blood pressure in lowering treatment to another risk factor for coronary heart disease that's independent of the impact of the treatment on blood pressure. So this is a hypothetical example. For example, maybe blood pressure lowering treatments have some off-target effect and they also affect your kidney function, and that's related to coronary heart disease. If that's true, if that last assumption is violated, then what you're estimating in your instrumental variable analysis is not the effect of blood pressure on coronary heart disease, it's the combined effect of blood pressure and whatever effect there is on kidney function on coronary heart disease, and that's not what you want. So important is that that's a very different source of bias to the residual confounding that we're worried about in conventional multivariable regression analyses. But also important that this is an instrumental variable analysis, the same as Mendelian randomization, and has exactly the same assumptions. The other thing that's important to know about instrumental variables analysis for me is, including Mendelian randomization, the calculation, the estimation of effect is very, very simple. It's much simpler than multivariable regression because it's a simple ratio. All you need to know is the effect of the instrumental variable on your risk factor and the effect on the outcome and divide one by the other. It's a simple division, simple ratio. So it's the effect of your antihypertensive on coronary heart disease divided by its effect on blood pressure. And here, because antihypertensives work on both systolic and diastolic blood pressure, and the two are related to each other, you're actually measuring the joint effect of both of these. So if your particular randomized control trial reduced blood pressure by 5 millimeters of mercury for systolic blood pressure, the higher one, and 2.5 for diastolic blood pressure, the lower one, and it also reduced coronary heart disease risk by 7.5%, but you actually wanted to model this or scale this or understand this in terms of a 10 millimeter of mercury reduction in systolic blood pressure or a 5 millimeter mercury reduction in diastolic blood pressure, the calculation is just really easy because you're doubling these, you just double that, and it's a 15% reduction. And you'll see with the later slides why I've particularly picked 10 and 5. Does that make sense to everybody? People comfortable with that simple analysis? Great. Okay, so the reason I was looking at 10 and 5 is because this is what I think is a really beautiful paper. It was published in the BMJ in 2009 by Malcolm Lawlor, Graham Wald, and J.K. Morris, who I don't know. And it was doing exactly that. It was saying, let's quantify the effect of lowering systolic blood pressure on coronary heart disease. Quite an important thing to do from a public health perspective. And how they did that was to meta-analyze in this instrumental variable way all of the published randomized control trials of blood pressure lowering treatment. So they weren't interested in the question of, does treatment by an antihypertensive reduce your coronary heart disease? They were interested in using those trials to say, what's the quantitative effect of reducing systolic blood pressure by 10 millimeters of mercury and or diastolic blood pressure by 5 on coronary heart disease? And these are the results. And one of the important things about these results is they separated it by different classes of antihypertension drugs. So thiazides, beta blockers, angiotensin converting enzymes, angiotensin receptor blockers, calcium channel blockers. These all work through very, very different mechanisms. Yet despite that, when they scale it in this same way of 10 millimeters of mercury drop in systolic and 5 of diastolic, you're pretty much getting a consistent effect of overall a 15% reduction in coronary heart disease. So what this suggests is that that violation of the exclusion restriction criteria, which is what econometricians call that third assumption, where you've got some other false pathway from blood pressure to coronary heart disease, is not existing, because it'd be very unlikely to get such similar results when you've got all these very different classes of drugs, which if they had some other effect, would be really unlikely to be the same effect and have the same magnitude and get you the same biased results across all these different classes. And this was what they concluded, and the paper has many more things in it which I think are really neat. But it was interesting to me that they used this term here. So they said all classes of blood pressure lowering drugs have a similar effect in reducing coronary heart disease events given the reduction in blood pressure, so given you've scaled them all the same, and so excluding material pleiotropic effects. And in genetic languages, this word pleiotropy is used all of the time to mean that you have a genetic variant that has more than one effect. And so when we switch this instrumental variable analysis to genetic variants, the term that's often used for that violation of that third assumption is a term called horizontal pleiotropy. So it's quite nice for me to see that it's not a language that needs to be restricted to genetics or make you anxious because it's a genetic language. It's not. It's simply saying that when you get something like this consistency, then it suggests that that other effect of randomization to hypertension drugs is not biasing the results, because you've got all these different mechanisms. And the other thing that's nice about this is if we can get genetic variants, so several different genetic variants that proxy for blood pressure, and like these are acting through different mechanisms, then in a sense we can use the same logic, assuming we have adequate statistical power. So this is just saying exactly what I'm saying. This is the sort of generalization of an instrumental variable analysis. So now I haven't put anything in here other than Z is the notation we're using for instrumental variable. That could be randomization to a blood pressure lowering treatment. It could be genetic variants that are related to blood pressure. It could be a whole host of other things that have been used as instrumental variables. It has to be related to X, the risk factor that you're interested in. There should be no link between your instrumental variable and confounding factors for the risk factor outcome association, which is Y drawn on here. And then lastly, this last, which the econometricians called the exclusion restriction criteria, and econometricians are the people that have mostly used an instrumental variable analysis. So if you're reading this literature, you may see this term. All it means is this third assumption that there's nothing linking your instrumental variable through to your outcome other than through its impact on the risk factor that you're interested in. And so, as I've said, this is what it might look like if we had SNPs. So the word SNPs is just single nuclear polymorphism. They're the type of genetic variant that we tend to use in Mendelian randomization. Mostly we use them as a 3-order categorical variable, going from having none of the blood pressure affecting alleles, one of the alleles, 2 of the alleles. So we often use a dose response across 3. Obviously with all things it helps if you know a little bit about what you're dealing with, but you don't need to fully understand genetics to grasp instrumental variable analysis in general or Mendelian randomization specifically. Okay, and again, as with the example I started with, where we used randomization to antihypertensives as the instrumental variable, and as in the BMJ paper example, the analysis is a simple ratio. So it's dividing the association of the genetic variants with coronary heart disease by the association of the genetic variants with blood pressure. So what I'm now going to do, if people are happy with that, and I see some nodding of heads, that's useful, is move on to this 2-sample Mendelian randomization and the use of aggregate data. And as I've said, I think it's important because its use is increasing and increasing. A little bit to me like when meta-analysis of randomized control trials first began. Very quickly people started to say rubbish in, rubbish out. And that referred to both the quality of the studies that you were using in your meta-analysis, but also to some extent on how you chose those studies and how you chose to put some in and some out and the impact that can have on your results. And the same very much applies here. So this was a commentary I was asked to do for the International Journal of Epidemiology, which was for one of their first 2-sample Mendelian randomization studies with aggregate data. And it was a study looking at the causal effects of adiposity measured by body mass index and waist circumference on a whole range of cancer outcomes. And it was by a very good group of authors, but my first reading of it, there just felt like there were too many mistakes, many mistakes, that the reviewers hadn't picked up. And so I suggested the editors go back with my comments and ask them to redo the paper. They said, no, we'd rather you put that in a commentary because we're seeing loads of papers like this and it's not good for the community and it's not good for Mendelian randomization. It's difficult. But for example, they went to the giant consortia where they got their genetic variance for body mass index and waist circumference, and they used the combined data for men and women for the genetic variance for body mass index and waist circumference for all of their outcomes, including breast cancer, which does occur in men but rarely, and prostate cancer, which definitely does not occur in women. And at a minimum, and that's one of the problems with aggregate data, maybe they only had the aggregate data on body mass index and waist circumference variance from men and women combined. But at a minimum then you need to discuss that. In actual fact, the consortia for body mass index and waist circumference have them separate by the genetic variance separate by men and women because there are some differences, and there were various other things. So it sort of raised concerns about this. And at the same time, the use of two-sample Mendelian randomization is increasing and increasing and increasing. So this is just from 2011, from when genome-wide consortia began to make all of their data publicly available. And this is one sample MR over that period increasing. This is the same, if not slightly steeper, increase in two-sample MR. And so consequently, what you're seeing is, as a proportion of one sample MR, a continuous increase of studies that are using this two-sample MR with aggregate data. And this paper, which at the time was accepted, it's now published in 2007 in the International Journal of Epidemiology by Fernando Hartwig, is well worth reading. Its main focus is on, when you're using this two-sample MR approach, making sure that you correctly harmonize the two samples, which is not a straightforward thing. We talked about it in my group today, but there isn't time to go through it here. So if you are interested in this, it's really important to work through the stages that Fernando suggests in this paper. Okay, so the basis of two-sample MR is simply that there is no reason why for the two bits of this ratio you shouldn't get those ratio estimates from two completely different samples, two independent samples. And there's actually some benefits to doing that. The biggest benefit, and the thing that's sort of pushing this most, is the sample size and the power that you can get from the available summary data now. It's leagues larger than any single consortia or than any single study. There are also some other issues. So what happened initially when people were using Mendelian randomization, which was the real extreme of overfitting your data, or what is sometimes called winner's curse, was that the genome-wide association study, to identify genetic variants that you would use for Mendelian randomization, was done in the same study that you then did the Mendelian randomization. So if I step back a bit, and again, one of the things we've been talking about in the group is, how do we select genetic variants to use in Mendelian randomization? We select them mostly, I would say 95, 99% of the time for published Mendelian randomization studies. The genetic variants have been selected to be those variants that are genome-wide significant in discovery samples, so large collections of cohorts and case control studies, and that replicate. So they're robustly associated. Their association is defined as robust based on statistics, p-values, f-statistics, r-squareds, because we're interested in their predictive ability for the risk factor we're interested in, and how strong that is in a statistical sense, but also how reliable that you see it replicated in different studies. So for example, for body mass index, the biggest consortia now, which is called the Giant Consortia, has identified 97 of these SNPs, these genetic variants, that are significant at p to at least 10 to the minus 8, and that replicate. So p is lower than some figure times 10 to the minus 8, and that replicate at that same value across samples. For blood pressure, the same criteria as 26 genetic variants. So that's how we're selecting the genetic variants. If you do one sample MR, and particularly if that's where you've generated your genetic instruments to use in Mendelian randomization, then you're overfitting the data and have got this winner's curse. And that tends to, in one sample MR, bias towards the exaggerated confounded effect. Same is true when you have what's called weak instrument bias. So where actually you've got a low R squared or a low F statistic in terms of the relationship between your genetic variants and your risk factor. That can occur in either one sample or two sample MR, but in one sample MR it biases towards the exaggerated confounder effect. In two sample MR it biases towards the null, and we're sort of conservative as epidemiologists and tend to prefer the bias towards the null. And then lastly, this two-sample Mendelian randomization approach, particularly with the summary data, lends itself now to some new methods that have been developed and are continuing to be developed for testing this concern we have about horizontal pleiotropy. And you might wonder whether it's the cart driving the horse or the horse driving the cart. It's a bit of both. So two-sample MR has become popular because of the massive sample sizes and because of the easy availability of the data. And these methods have then followed, but also they've become intuitive, because for those of you who work in meta-analysis, you'll be aware, for example, of the Egger test and some of the other tests that I show you that are used here. They're exploiting, they're developing methods that were originally available for use with randomized control trials, summary data, meta-analysis. So it's a bit of both. They can work in one sample MR, but they're not as straightforward and they don't work as well. Okay, so one slide only on two sample MR not using summary data, because of course it is possible and you can still gain some of those benefits of having the two samples separated, and so not having winner's curse, and again here if they're independent sample, any weak instrument bias would again be towards the null. The main difference is that you don't get the power. So the example I've given here is a made-up example, but this is the collaboration of genetic studies of cardiovascular disease from UCL, University College London, London School of Hygiene and Tropical Medicine, Edinburgh and Bristol. So it's a group of us who put all of our studies together to look at metabolomics and cardiometabolic disease. So we could run an association of known genetic variants that have been shown in genome-wide association studies to be related to testosterone. And we might have testosterone measured in about 10,000 of our samples. So we can run that and get the denominator in the UCLEB consortia. And then we can go to UK Biobank, which any minute now, everybody's waiting for the key to open their data sets. We've all been downloading it for the last 4 weeks. We'll have access to 500,000 people with genome-wide data. So we could then look at those same testosterone SNPs in UK Biobank and their relationship to coronary heart disease and use that as the numerator. And that would be fine. But in comparison with taking the testosterone GWAS data to cardiogram C4D, which is the biggest consortium of coronary heart disease, we have 64,000 cases of coronary heart disease in that consortia, whereas UK Biobank is hoping that by the end of this year they will have 20,000. So it's a threefold magnitude greater. So the main, as I've sort of emphasized, push for this two-sample MR using summary data is the accessibility of the data, which kind of fits with people like Ben Goldacre and others saying we can all compare results and people can challenge your results, do the same thing with the same data, but also it's got this massive power. So I'll make the slides available to anybody who's interested in them. In the interest of time, I'm not going to go through all of these. The limitations are primarily the limitations you get with any kind of aggregate data, whether it's randomized controlled trial data or genome data. So just briefly, I'm going to talk about what we mean by GWAS summary data, and then talk about the steps that we would go through in a two-sample Mendelian randomization using summary data, and then just give you some examples. So what we mean are all of the data that come from these consortia where what they are doing is basically 2 million, slightly more than 2 million chi-square tests. It's a great big set of chi-square tests. So again, nothing complicated. It doesn't matter what this is. This is often in the papers because it's not very interesting to try and present 2 million association statistics in a paper. Very few journals would accept that. They'll accept quite extensive supplementary tables, but usually no more than the variants that are genome-wide significant. So for example, 97 with BMI. And what you get in the paper is a picture like this Manhattan plot, which is all of those 2 million along the chromosome plotted against their log p-value. So these up here have got smaller p-values than these down here. And you take your genome-wide threshold and decide which of these are genome-wide significant. What the summary data produce are not data on individual participants, but they produce for each of the 2 million genetic variants the unique ID, a thing called an RS number for those genetic variants, the regression coefficient, the standard error, which of the 2 alleles that you can have at a particular part of your genome is being looked at in this particular genome-wide association study, which is the non-effect allele and the allele frequency. That's the kind of minimum data that you would want to be able to avoid problems with not harmonizing the data correctly. So this is just the top lot of things that I've pulled out of this giant consortia, which is where you can get GWAS data for these are waist circumference adjusted for body mass index. Personally, I prefer it if they hadn't adjusted for the body mass index, but you have no choice because this is what they provide for you. This is what the analyses were done with. They also have body mass index and height there. So this just goes on for 2 million. And as I say, you get what I've called A1, which is what they are treating as the effect allele, A2, which is the complement allele to that, the frequency of A1, the effect on waist circumference of whether you go from 0 to 1 or 1 to 2 copies of the T allele, and its standard error. And here they've given you the numbers that are included for each of these genetic variants. It varies because within the consortia, some studies use slightly different methods for collecting the genome-wide data to begin with and then imputing from those data to stretch it out to this total of 2 million or so. This, as I've said, there isn't time to talk about harmonization. It is a major problem or potentially major problem, and I'll show you why with an example later. But the reason you want at minimum this information is this is the way that the giant consortia have looked at these particular variants. There's no reason why in the coronary heart disease consortia they might not look at C as the effect allele. And worse than that, DNA sort of comes in 2 strands, and you can read forward or you can read backwards. And if you're reading backwards, you may not have T and C here at all. You may have G and A, but it might be exactly the same thing because reading backwards is the complement of reading forwards. But if you don't have some additional information, you've no idea about how to harmonize these with you can't just go, oh, let me look up this number and put in the beta and the standard error for that outcome. And as I say, Fernando's paper goes through that in really nice details. So just a quick summary of the sorts of data that are available. There's now 2,500 GWAS studies that have made their data publicly available in aggregate. And they cover about 1,500 different traits. So these is not by any means exhaustive. These are some of the consortia. These are some of the measures. So it goes from rheumatoid arthritis, education, blood pressure, anorexia nervosa, bone-related outcomes, kidney disease, all sorts of different diseases and potential risk factors. And again, just sort of summarising, we have gotten increasingly interested in these as risk factors, and we're now able to access genome-wide data for these to try and explore causal effects of these on various disease outcomes. So what do we actually do when we're doing an aggregate two-sample Mendelian randomisation? So in fairly simple terms, you would, if you're interested in systolic blood pressure as a risk factor, you would go either to the consortia database and or just look in the paper, because in the first sample, you select genetic variants in general that fit this criteria of being robustly related to your risk factor. So for sample 1, you're selecting out the genetic variants, a limited number, so for systolic blood pressure 26, that have reached some number times 10 to the minus 8 or smaller p-value significance and have been found in discovery data and replicated. So you have those 26, and then you might take that information and put it into an Excel spreadsheet or put it into your Stata or your R data file. Along with, so you put in the per allele difference in those SNPs and their standard error, together with these information that are going to help you in harmonizing those SNPs with the second sample, your outcome sample. So you then look up each of those 26 blood pressure SNPs in your second sample, sample 2. You then have to go through several stages, several steps, which, if you're interested in, have a look at Fernando's paper to make sure you have correctly harmonized and got in the same direction and actually measuring the same alleles for each SNP. And then it's a simple ratio of the two samples. So if this is what our data look like, and I'm putting this in only, these are made up data, but if this is the exposure allele and allele frequency, I'm assuming that we've harmonized it with the outcome data. So you have one regression coefficient for each of these genetic variants. These are not any particular, this is what the identifiers look like, but these are not related to anything in particular. You have one beta for the gene with exposure and its standard error, one beta for the gene with outcome and its standard error. I didn't bother putting in the standard errors. And then you just divide this by this and you get a set of ratios. The standard error calculation for the ratios has to take account of both of these standard errors. That's doable, and there are packages for doing that. And then it's a meta-analysis. Similar methods to a meta-analysis for randomized control trials, but different in that meta-analysis for randomized control trials are on independent study samples. What we're looking at here, what we're trying to get at, is independent genetic instruments, and that's what we do the meta-analysis on. So then different forms of meta-analysis can be used, and again, many of these, if you do meta-analysis on randomized control trials, will be familiar to you. You can do either a fixed or a random effects meta-analysis of those Wald ratios, and then you can use Cochrane's Q statistics, the I-squared statistic, leaving one out and redoing the meta-analysis again the same way that you do with randomized control trials. And they're testing heterogeneity In the same way that I talked about the independent blood pressure lowering drugs having different pathways, if there were heterogeneity between them, you'd be worried that at least some of them are pleiotropic. If there's not, then that gives you some reassurance that they are all estimating the same causal effect. And so exactly the same applies here when you use genetic variance. You can do an inverse variance weighted meta-analysis, and I'll show you an example of what that is in a second. That approach assumes that there's no horizontal pleiotropy, so they assume that the last criteria is not violated. And then there are these newer methods, particularly MR-AGA has a very clear relationship to inverse variance weighting. It allows you to relax this assumption, but has some other assumptions of its own. The nice thing about these three in particular is they have quite distinct and different assumptions. So again, if you do these and you see things looking the same, you feel more confident that that's the true causal effect. So inverse variance weighted meta-analysis, this was new to me when not new to me, I'd seen it done in randomized control trials before. I think I'd done it myself in randomized control trials. What I'd never realized was quite what it was doing. What it's doing is a kind of regression analysis to get to the Wald ratio. So this is actually an inverse variance weighted Mendelian randomization study. So these are individual, independent genetic variants that affect low-density lipoprotein cholesterol, which is on the x-axis, and it's the weighted regression through their mean effect on low-density lipoprotein cholesterol and their mean effect on coronary heart disease. But you could imagine this was a set of randomized control trials of different doses of statins or different treatments for reducing low-density lipoprotein cholesterol. The thing that I'd never realized before about this approach is it's always orientated this way so that it's a positive slope. Whether you're interested in a positive slope or not, that's how it's orientated. And that's done because the regression line is forced through zero in inverse variance weighted meta-analysis. So it's accepting the assumption that if you don't have genetic lowering of your low-density lipoprotein cholesterol, or if this was randomized control trials, if you weren't allocated to the intervention to reduce your low-density lipoprotein cholesterol, then there could be no effect on coronary heart disease. So it's totally accepting that there is no violation of that third assumption, which is why this can only be correct and unbiased if there is no pleiotropic effect. It looks here, certainly for these, there's a sort of line around these ones down here, that going through the zero is not unreasonable. But this might change things a bit and make it sort of not quite zero. And what MR-EGA does is exactly the same as an inverse variance weighted meta-analysis, makes them all orientate to a positive slope. The only thing it does different is not enforce it to go through zero, which is kind of quite neat, because then what you have here is an intercept, which is the sort of overall measure of any pleiotropic effects across individual independent SNPs in Mendelian randomization or randomized control trials. It doesn't necessarily point out which of the trials or SNPs is the one that's causing this pleiotropy, but it warns you to its presence, and it gives you a new slope for which you can relax that assumption. So this is almost like a sort of pleiotropy adjusted slope. Okay, so this is, again, I'm not going to talk through this. This is just a summary of the differences between one sample and two sample MR, both in terms of the kinds of analysis that we would do and some of their strengths and limitations. This is a summary of the methods and how we use these methods in two sample MR. And what I'm going to move on now is to show you some examples of where these have been used. So this first one, again, this is, to my mind, a really nice paper by Brian Variance and colleagues where, again, they were asking this question, what's the magnitude of causal effect of reducing, here they were focusing just on systolic blood pressure, of reducing systolic blood pressure on coronary heart disease. And they looked at it in 3 ways. They looked at Mendelian randomisation analysis. Both of these top 2 are Mendelian randomisation analyses done in the same data sets. One uses 12 of the known 26 genetic variants, one uses 25. The next is the Oxford's group large individual participant meta-analysis of cohorts, so multivariable regression analyses. And the last are the summary results from the randomized control trial meta-analysis that I showed you at the start, where they were using an instrumental variable analysis. What you can see from all of these 3 methods, each of which have their own individual different sources of bias, is they all suggest that lowering blood pressure lowers your coronary heart disease. But there's heterogeneity. So in comparison to the Mendelian randomization analysis, the prospective cohort multivariable regression analyses are weaker. The randomized control trial analyses are weaker still. So one question when you see this is to consider whether there's some bias in this that exaggerates it or some bias in these that sort of push them closer towards the null. But the other thing that's important to consider when you're doing this sort of triangulation work is the extent to which these different methods are measuring different exposure length to the risk factor. And that's what they showed in this paper, or strongly suggested, when they look at the randomized control trial was followed up for about 5 years. The prospective cohort study about 13 years, whereas your genetic variance, your susceptibility to genetically lowered blood pressure is with you for life, and the average age of the participants here was 55. So not only does this work together say that lowering blood pressure lowers your coronary heart disease, and because it's been looked at in different ways with different methods, but also different approaches. So one is measuring randomization to antihypertensive treatments. One is whatever has caused you on average to be lower in blood pressure than somebody else in an observational study, and the other is genetically lowering. It suggests that lowering your blood pressure by any means will reduce your coronary heart disease, and more so, the longer you have lower blood pressure, the lower your coronary heart disease will be. Sorry, I didn't mean to go back there. Okay, so another example, and this is Maria Carolina's work, really nice 2-sample MR looking at the effect of adiponectin on coronary heart disease. And here we again took a number of different approaches. The reasons for that were we had data on the risk factor part of the ratio from the adipogen consortia. So we're able to look at genetic variants that have been shown to robustly associate with adiponectin in 46,000 participants. So again, this is another example of the scale and the sample size you have here. We looked at coronary heart disease outcomes with 2 data sets, one which was this cardiogram consortia with 22,000 coronary heart disease cases, but then they joined with another consortia called C4D, which takes it up to 64,000 coronary heart disease cases. For adiponectin, there are some genetic variants that are in the gene region that codes for the protein that is adiponectin. So there are variants in there that are independent of each other, but are all relating to coding for adiponectin. So for those, we think it's very unlikely that there will be pleiotropy. Those genetic variants may relate to other outcomes, but they'll be downstream of the protein that they're coding for. So that wouldn't be, it would be what gets called vertical pleiotropy. It doesn't matter, it's not going to violate your assumptions. So we did some analysis where we just looked at the 4 genetic variants that we had in that gene region that we know are coding for adiponectin. And that we called conservative because it's conservative in terms of statistical power because there are just 4 genetic variants there. And then we did a more liberal approach where we included all 17 genetic variants, those 4 in the adiponectin gene, but also any other genetic variant that had been shown to be genome-wide significant and replicated. We knew that some of the genetic variants in those additional 13 also have been shown to relate to glucose type measures and lipid type measures. So it's possible that they would violate either the second or third assumption. So it's possible that glucose and lipids and body mass index was the other one are confounders for the association, the observational association between adiponectin and coronary heart disease. And if the genetic variants relate to those confounders, then you're violating assumption 2. It's also possible that they're independently of adiponectin related to those outcomes, in which case you might be violating assumption 3, the exclusion restriction criteria. And then lastly, it's possible that they're on a causal path. It's possible that adiponectin causes variation in glucose and lipids, and that's part of how they affect coronary heart disease. So we wanted to try and unpick that, and we did that by, for these, all the 17, the sort of liberal approaches, looking at how they and the conservative adipogen ones that we knew were just affecting adiponectin related to these outcomes. So this is the first set of results where we're looking at these potential pleiotropic or confounding effects. If the truth was that adiponectin caused variation in glucose and lipids and body mass index, and they then caused coronary heart disease, so they're mediating, it's all part of the adiponectin effect, we'd expect these genes that directly code for adiponectin to be related to the confounders. And by and large, there's not much evidence they are. If these are either pleiotropic or confounding, then we'd expect the liberal, the non-coding variants to be related to some of them. And by and large, we can see that they are. So in the final analysis, and there's not time to go through this, but it's an illustration, I think, of how you can pick and choose what you present and what you do. So what we have here is shown everything that we've done in the smaller sample with 22,000 cases, in the larger sample with 66,000 cases, doing the conservative inverse variance weighted, where we think there's no pleiotropy in both samples, doing the liberal but with no adjustment for potential confounders, doing the liberal where we do adjust for potential confounders, and doing the MR-EGA. The pattern's broadly similar in both, but when we have the larger sample, it's much clearer, if you think of these two as our most robust analyses, that there seems to be no causal effect. The MR-EGA, if anything, suggests a positive, and the hypothesis is a negative effect. It's not possible to go through, but we think that's because there it's violating one of the assumptions of the MR-EGA thing. So I'll finish now. So we could have presented just this and said, yeah, this agrees with a protective effect, which many of the reviewers would have been interested in. We published all of it and got a very nasty editorial, but I think we did the right thing. I'm not going to go over any of this. I will make the data available, except this. I think this is important to see. So these are 2 papers that have now been published for some time, using exactly the same data sets to ask exactly the same question. What's the causal effect of CRP on schizophrenia? And one tells you it's positive and causal, if you believe all the assumptions of Mendelian randomisation, and one tells you it's negative and causal. And if you get the chance, as I've mentioned several times, to read Fernando's paper, you'll see that interestingly, there are errors in both of these. There are errors around the whole harmonisation process, but particularly in this one. This one is incorrect. This is nearer to the correct answer, but it's worth having a look at Fernando's paper. So that's the correct answer, done very carefully with a lot of scrutiny. Okay, lastly, we have generated, and we is not me at all, I'm not clever enough to do this, but we put together the 2,500 aggregate GWAS data that are available into this MR base, and behind the scenes, all of the R code that you need to harmonise data sets and to run MR-EGR, median methods, anything and everything you want to do. It produces nice graphs. It will show you the R code that's being used. It will give you the citations. So when I said you could go out of here and do a 2-sample MR in between half an hour and 2 hours, you could by registering on here and doing this. I think you still need to engage your brain and think about just what's going on and why you're doing these analyses. But if you're interested in knowing more about these, and I've said I'll make the slides available, then this is the paper that describes it. This is the actual interface that you can go and register on and play with those. And as always, the group that have put that together are very keen for feedback on how it's working. And so with particular thanks to Phil Haycock, who teaches on the Mendelian Randomisation course that I run and helped me with some of these slides and has been completely instrumental to the MR base interface, and other colleagues and friends who are too many to mention. Thank you, and sorry for running over.